And it basically is created when your rod, the diopter, is excited at a relatively low frequency, but over the course of the life of that diopter molecule, it may produce some of these molecules, and then they essentially are non-functional. They are chemically damaging. They're divergent molecules that get segregated as you age into cells right behind the photoreceptor layers, and it's hard to read layers that they can do. So we think this A2E molecule, and there's a lot of evidence that it can be a stressor in early damage to this RPE cell layer and in age-related maculopathy, which is the most common cause of blindness. This is a big problem. And the thing that attracts me to my sort of theory is that if the theory is right, there's a simple solution to protect your eye. That if there's a spectral balance with aging, as your retina is getting too little blue light or more green light, then the balance, appropriate balance relative to the blue light, you can protect the retina with sunglasses. And the main damage for any of these photochemical processes would be when you're outdoors in sunlight. So it's just not just giving people sunglasses, but the right spectral rebalancing. I have prepared here this sort of, we designed for clinical studies where we could actually compare one eye versus the other with two different hypotheses. One that blue light damages, and the other that actually you need blue light relative to non-green light. More generally, and this is what really why I feel in good conscience I can work with mathematicians here, is that there are a whole wide range of problems in retinal disease and retinal pathology that are chronic diseases that develop very slowly, like age-related macular degeneration, for example, diabetic retinopathy, that are really incredibly large burdens on our population. Vision is highly prized, and as we have an aging population, these are really critical medical problems. And if we were better at imaging and analyzing those images so that we could look at the very earliest pathological changes, then we might be able to test out rapidly things that might be beneficial and come up with disease prevention strategies. Right now, the standard methodology is that in age-related macular degeneration, you take people with already fairly moderately advanced disease that have a 20% chance of going to visual loss in 10 years, and you study a couple thousand every year for 10 years, which is a very expensive, costly study, and also maybe well, after you're dealing with very advanced pathology, simple interventions may not work. So just to give you a little bit of biological background, the RPE layer is at the very back of the retina behind the photoreceptors. And they're essentially a nurse-like layer. They are ensuring the health of the photoreceptors and the nearby overlying retina. And one of their functions is that the photoreceptors in their outer segments have pigment molecules. They're essentially like the grains in your film camera, but they're actually regeneratable, so that the rhodopsin protein molecules has a chromophore bound in it, and light excites it and undergoes a conformational change, and that starts a cascade of signaling that light was incident. But that molecule, the chromophore, the rhodopsin, the retinal, actually changes its conformation, which is what when it was excited, and then it actually gets separated and makes a circuitous route through the RPE cell and gets regenerated. This is a normal visual cycle. If you have too much of that transretinal, which is highly reactive in the outer segments of the photoreceptors, particularly the rods, there's a cross reaction that forms a fluorescent product that is not digestible, and it actually accumulates as you age in the RPE at the back of the retina. So there are two things about this. The clinicians are very interested because now we have a way of imaging the back of the retina in the RPE because it's actually emitting light when you put in exciting light. And so it's very sensitive to the structures of this very critical cell layer, and it's also sensitive to any overlying absorbing molecules in other cellular structures. So this is just a schematic of this sort of visual cycle that I was trying to tell you about. There's rhodopsin in the rods, that's the photopigment in the rods that's sensing light. It undergoes a conformational change with visual light, the transretinal in it. The cisretinal undergoes this conformational change. And then this actually gets uncoupled from the protein molecule to which it was bound covalently and undergoes this sort of path of a series of enzymatic reactions where it then is restored to the original crooked retinal that then binds back to the rhodopsin. And so that's a normal cycling mechanism. Typically, that might go on for about 10 days, and after 10 days, those molecules are pretty much spent. And this membrane section of hundreds of these little stacks of photoreceptor layers actually gets shed in the RP phagocytosis and starts to degrade it. And it's that it degrades almost all of that material, 99% of the material, but there's a small residuum that it can't digest with its package of enzymes, and that just accumulates in a granule. And these granules are highly fluorescent. That means we can put in short wavelength light and get longer wavelength in the first photoproduct, is A2E. We get, if we put in light in the blue at 430, we get out red light. And there are, in this pathway, a number of different molecules. There's a precursor, which is in the outer segments, which actually has a longer excitation wavelength. And then what happens in the granule in the RPE is, over time, as blue light is incident on that granule, it gets absorbed. These are the blue end of the spectrum. And it activates singlet oxygen. So it actually, the excited state molecule transfers its energy to oxygen, and singlet oxygen is very reactive, but very short-lived. And so it just reacts with carbon double bonds that are in that granule. It turns out there are a lot of carbon double bonds in this transretinal. And so that actually is a way that these retinoids that are formed and accumulated are actually being reacted with. By themselves, they're almost chemically inert. But if they're reacted with, then they can cross-react, and they can essentially precipitate out where they can't go into solution in the end the RPE cell. That, in ways, is my hypothesis. We have some evidence for this, and we can do studies using spectral microscopy. And so this is a mouse model. A couple genes are knocked out, and those genes mean that there's more of this A2E molecule that's actually accumulating in the RPE cell. And this actually replicates pretty well if you keep the mice alive long enough to all of or a number of the end stages of age-related macular degeneration in humans, including neovascularization of fluoride. And here's an example of 1 year there's a large disruption of the RPE. It's highly damaged, and the overlying retina is starting to be damaged too. If you look at in the spectral confocal, we can, in a microscope, we can actually have fairly detailed spectral information. Not hyperspectral, but fairly detailed. Part of this is in a microscope, we can very efficiently put in light and collect the fluorescence through a very large angle. We can't do that in the retina. So we intrinsically can get about 100 times more photons per photon in a biological microscope sample than we can in human retina. So we have a lot of signal to work with, and you you can do these studies, you're also not as concerned about causing some low-level injury, photochemical injury, that you would be when you're dealing with patients and trying to use them non-invasively. So that just is a point of reference. We do have some spectral information. It suggests that the molecules in the mouse, which has very low ambient light levels, it produces the A2E, but it really doesn't change over a year. It's still largely this A2E molecule. In humans, where we get more chronic light exposure reaching the retina, we actually see that there's a shift in the wavelength, and it looks like there's a photochemical change, which is what I was suggesting in the previous slide. I don't even show it, was that this is actually has a substantial portion of oxides that have actually been created by that singlet oxygen like fusing rings. You can do acute experiments like this, where you take cells in culture and you feed them A2E, and you expose them. You can see how much light it takes to do this conversion from A2E to the oxide. And that's a process, I think, that's naturally occurring in the human retina, but very slow. In the microscope, we're doing it very rapidly, but in humans, it's taking months or even years, depending on how much blue light reaches your retina. You can do exactly the same thing in the mouse. This was the 2 photons that causes some damage, but basically you can also bleach the A2E in a mouse eye that normally doesn't get much blue light, at least over the year of the slide. So what's happening in our eyes as we age, that you have a large, thick lens, which as it ages, it starts to get a discoloration. It starts to become yellow. There's some chemical products, and they probably are photochemically active, so there's probably a little bit of a feedback. So you get this progressive yellowing in a population at large. Some people that have a nuclear cataract who tend to be even more extreme than their common age cohort, and somebody that had a very robust lens might not be quite as yellow, but this is happening in everybody. So as you age, there is a shift, so you're getting less blue light and more green light, relatively, getting less of both, but substantially less blue light compared to green light. And here's the spectra, the action spectra, for the 2 processes I was interested in. Activating rhodopsin with green light is what, when you're out in bright daylight, is actually producing this A2E chemical, so that has to do with production rate. And the short wavelength, violet light, blue light, is the one that's actually causing the chemical conversion in the lipofuscin granule in the RP. And we're postulating that is what's critical for detoxifying that molecule. This is in cell culture, we can actually look at the doses, and we can relate those doses to how much light you would actually get to have that bleaching. What is the total blue light flux or spectral flux that would cause the conversion of A2E to the downstream products that we think are safer? And so I developed a complicated biophysical model based on a lot of data that people know about kinetics of a variety of processes and a little bit of hand waving. But basically, if you just look at this natural process that's occurring in everyone, not just in people that might have AMD, that as you age, because the lens is yellowing, you and other factors, that pupils tend to get smaller with age, you tend to get less A2E being produced, less rhodopsin activation, even if you had constant light exposure, same regardless of your age. But because the blue light level, this was basically being created by green light, the blue light level was so much lower, you actually, this accumulates, the time constant for its bleaching increases with age, and so even at a lower production rate, you actually get almost a linear increase in the amount of this fluorescent product. This sort of agrees with the limited clinical data that we have from Delory, where he looked at the red fluorescence, which he attributed to this A2E molecule, in a general population that increases linearly with age. So, and the other little point here is that if in this model I just assume some sort of constant daily exposure, fluctuating daily exposure to light, and if I increase the number of hours in bright daylight over a very broad range, sixfold, it really didn't matter. So it's the spectral balance, not the intensity of light, that seems to be important in my model. So before I started to talk to the Norbert Wiener Center, we did some preliminary studies with these bicolor glasses. And just if I wore them for a week at the beach, this is an autofluorescence image of those fluorescent molecules at the back of the retina, taken non-invasively. And this is sort of a color rendition, so it's a little bit easier to see. But for the eye that had the rods, which are largely in this region out here, when those eyes that had the rods being protected with a vermilion filter actually did not seem to have an appreciable change in the amount of fluorescence that we detected after just a brief period, whereas the eye that we had the yellow filter seemed to increase substantially. And we reversed this. But these are absolute measurements of intensity based on clinical instrumentation, which is intrinsically fraught with our inability to be sure that we're not just dealing with some other instrumentation artifact. This is, at least when we looked at the pattern, this eye was the one that had the increased exposure to green light, and therefore it had an increased production of the A2E pathway molecules. And it pretty much, if we've mapped out the fractional change, it follows the distribution of rhodopsin or rods in the retina, which seem at least to be self-consistent. But at this point, I started to say, well, how would I really prove this analysis? And I wanted to do studies in elderly patients that had just undergone cataract surgery, so I didn't have to worry about the lens attenuation and confounding of my measurements, and then give them these bicolor glasses. And so we set up this multidisciplinary team, and then I recruited people from the Norbert Wiener Center to see if they could help bring some image analysis expertise to see if we can be more clever in terms of how we limit our, what we're calling multispectral retinal images, to come up with things where we can actually better identify the compositional balance of these pores. Does it really shift as my model would predict under the spectral imbalances that we can create clinically? And whether we can then go farther than that and to start look if we perturb or say reduce the production of this A2E molecule, could we actually see that lesions might reverse themselves? So we would have measures of very early disease that we haven't presently been on the clinical screen. People generally, we're talking about preclinical pathology, so very minimal pathology that are decades away from actually causing visual loss, if at all. So these are things that clinicians have found difficult to study. Well, to start off with, it was partly to ingratiate ourselves with the Eye Institute investigators who are doing a large clinical study where they're trying to see if they give dietary supplements of lutein and zeaxanthin, which are, again, retinoid molecules that actually are the molecule that causes this dark spot in the center of your retina where the fovea is. And what it is it tends to go into the cell membranes of the axons of the photoreceptors and principally the cones. So this is, in this region, you have a very high density of cone receptors, and their axons run across the surface of the fovea. That's where you have your high acuity vision. And those molecules, the macular pigment molecules actually reside right here in the nerve axon membranes. People think that they're photoprotective or antioxidant molecules, and so that was the idea for giving dietary supplements. They gave dietary supplements, but they didn't have a reliable way of actually measuring whether or not the supplements were causing any changes in the concentration in the retina. And so that's what we tried to develop, a robust technique where they could use a standard fundus camera with the right kind of filters to look at the autofluorescence of the RPE and how it's attenuated by this overlying layer of macular pigment. Macular pigment has an absorption peak at 460 nanometers, and therefore, if you excite at 460, it filters the light before it reaches the RPEs. You get a diminished intensity here in the center where its concentration is the highest. And if you excite at 560, where it doesn't absorb appreciably, you don't have that attenuation. So if you basically can normalize these images, you can form a ratio and take the log, and that really is the attenuation factor. It's associated with the macular pigment, which is related to its concentration times path length, but this macular pigment is all above the fluorescent layer, so it becomes a reliable measurement. And that's essentially a two-dimensional map of the projected optical density of this macular pigment molecule. We did, we sort of hand-built, modified an existing instrument, and were careful to reproduce some of the parameters, and then did a small cell study in normals, and we saw very good correlations between our peak values and the values that were determined by a psychophysical technique, which is the only quote standard technique, but it's very difficult to do on patients to determine macular pigment density. And so it looks like we have a fairly quantitative measure, but then we wanted to go from this special instrument to just a conventional fundus camera in the eye clinic, and that's where we started to have a little bit more complications, and we're trying to come up with a robust algorithm to make that really work well. Well, this is one example where we looked at macular pigment distributions in an entirely different disease population. The Eye Institute had recruited a number of individuals with macular telangiectasia, which is a disease that's not very well understood, but one of the long-standing first findings is there's a region close to the fovea that starts turning bluish in a fundus photograph. So this is just putting in white light and looking at a reflected image. And you can see that bluish region, which is not apparent in this image, would be related to new vessel growth. So there's more hemoglobin in vessels at the surface, and so that's absorbing all the visible light, but the blue light is scattered more, so it looks sort of bluish sheen. In this patient who had that, very clear signs of macular telangiectasia in the other eye. It's a genetic disease that occurs just with some delay in both eyes. This eye was perfectly normal by measurements of either the fundus camera normal imaging or doing fluorescein angiography, where they inject contrast agent in the vessels at fluorescence. That's one reason we have cameras routinely that measure fluorescence. Is a standard for measuring vascular abnormalities in the retina is to inject a fluorescent dye in the bloodstream that circulates. And you can see the vessels light up. In this case, there was not a dense residual place where new vessels were leaking fluorescein. So this was a normal fluorescein angiogram. This is an optical coherence tomographic image, which actually is looking down on the fovea, and here the contrast here is just backscatter. So the retina is very transparent. And only when you get to the bottom layer with RP is it becomes less transparent. That's because the photoreceptors are right down here, so you want everything above it to be transparent. But there is some reflection in a very sensitive system like an optical sonography system. But that gives us some measure of the Z dimension of these images. Well, these are two autofluorescence images in two different spectral bands. This is blue and yellow excitation. So if there was macular pigment here, we would expect them to be markedly different. And in fact, when we compute the macular pigment map, we see right in the fovea and the parafoveal region, there's almost no macular pigment. And so this paper we were saying was that there's evidence now that maybe this is the earliest stage of disease, that there is something going on with the photoreceptor axons where they're losing the macular pigment under stress, and that's starting a whole process of then neovascularization of the retinal vessels nearby. The point being that this is kind of an example of how we use our macular pigment mapping. I wouldn't really tout very strongly that this is absolutely quantitative, but it's certainly relative. We can see the nice hole. Using conventional fundus camera images in the eye clinic in a rather simple multispectral protocol, and that it's a way of looking at early disease progression that really wasn't available before. Now I wanted just to reinforce some of the quality. This is a specialized instrument, a scanning laser ophthalmoscope, where we actually have a laser spot that scans the retina. It actually puts a lot less light into the retina as a fundus camera, but because it's a single spot that moves across the retina, we can reject a lot of sources of background that we see in the fundus camera. We can reject the signal from lens fluorescence that might be reflected by the retina, and we can reject any diffuse scatter, the fact that we're exciting fluorescence. This is actually the inverted image, so the dark spots are really where the fluorescence is the brightest. But I did this inverted image because it shows very clearly the vessels. So these are the retinal vessels, large vessels, and I'm not sure whether you can see it very distinctly, but out here you can clearly see there's fine structure, and this is actually showing that there's contrast due to the microcirculation of the retina. So in itself, this is clinically rather interesting if we can characterize this fine, very fine structure density, which you may not be able to see from there in here, and also look for regions where it might have dropped out because of some vascular change, or in places where there's new vessel growth, both of which, say, occur in diabetic retinopathy as a common indicator. But in this case, we did it without injecting the contrast agent. We really are using the fact that the back of the retina behind the vessels is lighting up with the fluorescent molecules at the back in the RPE, and the exciting light had to go through the vessels, and that causes a local contrast. So in the center region where we measure the macular pigment, there are obviously two components. There's one from the fine vessels, the hemoglobin in the fine vessels, which also absorbs the blue light, and there's also a macular pigment component. As you go farther out, when there's no macular pigment, the contrast gets better. When you're in this region where there's some macular pigment and some vascular retinal microvessels, it's a combination, so we have to correct for the vessel component. And in the very center, where it's the densest, there are actually no retinal vessels, so that's a truer value. In comparison, if we look at the same image with a green laser, which is a very special, there are only a few of those in the world, a green laser confocal scanning laser ophthalmoscope, we see the vessel structure, but we really don't see the macular pigment structure. So it's the ratio of the two that would give us a reasonable indication of the macular pigment distribution. But again, the vessels itself contrast of anything that's overlying the underlying fluorescent layer as well. What we're now have implemented in the eye clinic what we call a multispectral standard protocol, where we have 6 different fluorescent wavelength pairs, and we create a series of images. So this is essentially the blue confocal scanning laser ophthalmoscope and the 2 fundus camera images. We have 5 other fundus camera images, and then we take repeat measurements of those. And these are the inverted images in a patient that has rather advanced AMD, although still good vision in this eye. And the interesting thing is that you can see the vessel structure in the scanning laser ophthalmoscope, but using the fundus camera at the longer wavelengths, we see a lot of lesion contrast. So immediately that's telling us that by going to multiple spectra and combining the images in some way, we will have detailed information, spectral and spatial information about specific lesions. And that's what we're really trying to develop analysis tools to characterize those lesions, classify them, and then follow how they progress in size or frequency. And as you can also see that this retina is full of pathology that is really most apparent if you look at the autofluorescence images rather than just looking at a reflectance image where you wouldn't see anywhere near this contrast of detail. We have a couple problems that I'm just proposing right now that we're still struggling with. One is that the commercial fundus cameras have an optical design element that intrinsically has a non-uniform illumination. But that non-uniformity has to do with the specific fixation point of the patient, the aberrations of the lens and the optic media, and exactly how the photographer, the crispness of the focus. So it's not something that is that well defined a priori that we could just correct for. One measure of it that we've been doing is we take repeat images, like this is taking 4 images, essentially identical images, with slightly different focal point, fixation points. So we move the fixation point around on the patient, and then we do a PCA analysis. We can see that here's the common mode image, and then this is the variation that has to do with the illumination non-uniformities, but it's kind of a 4-fold problem. We really would like a way, and this is in the normal eye, to take those patterns or some equivalent to how do we actually remove this illumination non-uniformity to get a better quantitative value for all the pixel intensities in our images. One solution for looking at another problem is the fact that in the scanning laser ophthalmoscope we have confocal rejection of the lens fluorescence that might be reflected by the retina, and we have rejection of local scatter because we're only illuminating a spot and sensing from that spot in the instant, a very small spot. So we don't get contributions, whereas when we do the fundus camera, we're illuminating the whole field. And we're only collecting directly about 1% of the light that's coming out, so the rest of the 99% is bouncing around the retina. So by doing some sort of comparison of the 2 different instrument modalities, we can come up with patterns that are suggestive of the contribution from backscatter of the lens fluorescence and scattering of the background or diffuse, like diffuse skin fluorescence in the back of the retina when it's coming on its way out. And maybe that's a way to correct our images. Anyways, what we're working for is to integrate our fundus camera and the confocal scanning laser ophthalmoscope. They're different modalities. The latest confocal scanning laser ophthalmoscopes are integrated with the 3-dimensional optical coherence hologram, so we get 3-dimensional morphological data, not chemical data. And we're trying to develop a robust way to interpolate and register these complex image sets to correct for the instrumentation errors and non-local background subtraction, and then generate an improved understanding of the molecular and morphological sources of the differential contrast, and then use those on the corrected images to come up with a way of characterizing the chemical and structural composition of early lesions that we might then be able to follow disease progression. Thank you very much.
Any questions or comments?
I have a question. Are there epidemiological studies that are based on lifestyle that would correlate exposure to natural light or other kinds of things, incidence of disease like AMD?
There have been a lot of studies, and they're very equivocal. These are large epidemiological studies. Generally, there's not a strong finding, and that is something that actually argues against the conventional wisdom that blue light might be damaging, right? Because there's not a relation there in terms of early exposure to blue light. The other thing that the epidemiological data doesn't show is after cataract surgery, where you remove the blue absorbing lens and put in a clear intraocular lens, that there's not a dramatic progression of age-related macular degeneration. Our model basically says, yeah, that's right, because actually you're lowering the A2E level, and we think that's the stressor. The conventional idea of blue light toxicity has a hard time explaining why we don't have an epidemic of AMD, rapid progression after cataract surgery. I'm sorry?
So maybe we can leave the trying out the sunglasses for the break part. Thank you, but very